Hello students, today we are going to discuss in detail about eukaryotic replication. First of all, we would like to know what is the replication. Already we have seen in the case of the prokaryotic replication, which is the process by which the genetic material is copied. In the case of the eukaryotic system, it is very complex when compared to the prokaryotic system because the DNA is wrapped around histone and is compactly placed in the case of our nucleus. And in the case of the eukaryotic system, especially in the case of human being, we have 23 pairs of chromosomes, that is 22 autosomes, and 1 XX in the case of the female and XY in the case of the male. So how many number of base pairs are found and how many cells are found in the case of our system is nearly 100 trillion cells are found in our body, and we have 12 billion base pairs are to be copied. This is a tedious task when compared to the prokaryotic system for passing to the next generation. That is, it's a tough job where the DNA material of every chromosome has to be copied without any error, because if any mistake arises, that resulting in deleterious impact on the function of the protein that leads to some genetic disorders. So normally replication in the case of the eukaryotic system occurs simultaneously at a various point, like in a NH the construction of bridge is going on at various places now, so that they can finish the task of completing the completing the bridge work simultaneously, and to ease the connectivity of one place to the other place for the transport purpose. Same manner here also we come across the replication is started at various places. We call it as multiple origin sites or multiple replicons are available, then the process of replication going. First we would like to know what are all the process step by step in the case of the eukaryotic replication. The eukaryotic replication is not as much easy like your bacterial replication. The distinctive feature of the eukaryotic replication arises from the differences in the chromosome structure as well as the enzymes, proteins that are associated with the process of replication. And the eukaryotic system, already I told you, it is a multiple chromosome. It is very larger in size when compared to the bacteria, yeast, fungi. And moreover, in the case of the eukaryotic DNA, it is linear in nature. Like the bacterial chromosomal DNA, it is linear in nature, but in the prokaryotes, that is bacteria, the DNA, that is chromosomal DNA, is devoid of histones, whereas here histones are added to the chromosomal organization in the case of our system. So normally the DNA is wound around the histone. You know very well the level of organization of the DNA, how it is wrapped around the histone protein, what is 20 Armstrong and 200 Armstrong region of your histones, how it is placed compactly in the case of our nucleus. That has to be removed during the process of a replication. Without removal of DNA histones, it is not at all possible to carry out the process. So we call it as de-histone, that is removal of the histone is taking place during the replication process, or we call it as unwrapping of histone has to be done. So we must know the enzymatic machinery which are necessary for the process of our replication. So unlike our prokaryotic system, we have only 3 polymerases, 1, 2 and 3 majorly that is involved, but in the case of the eukaryotic enzymes, 5 common enzymes are available for the process of a replication, that is DNA polymerase alpha, beta, gamma, delta and epsilon. The alpha is responsible for synthesis of our RNA primer. It is similar to primase which is available in the prokaryotic system. It is responsible for production of primer in both leading as well as lagging strand of the DNA, whereas DNA polymerase beta is responsible for our DNA repair mechanism, just like DNA polymerase 2 of the prokaryote replication, and DNA polymerase gamma is responsible for mitochondrial DNA replication. This is the only circular DNA which is available in eukaryotes. So then chloroplast as well as mitochondria alone as circular DNA. In the case of the animals, the mitochondria containing circular DNA is a semi-autonomous. It is not depending on any protein from the nucleus, but it independently replicates. For the replication of the mitochondrial DNA, we require DNA polymerase gamma, and further DNA polymerase gamma is also responsible for proofreading function. And DNA polymerase delta is a nuclear DNA that is responsible for is found in the nucleus and is responsible for leading strand synthesis, and it is a leading strand enzyme is DNA polymerase delta. It also has proofreading function, and DNA polymerase epsilon is responsible for the production of your lagging strand synthesis, just like Okazaki fragments. Here also in our eukaryotic system also fragments of DNA is synthesized in the lagging strand template. This is similar to the action of our DNA polymerase 1. DNA polymerase delta is similar to the action of our DNA polymerase 3 in the prokaryotic system. So this is the list of enzymes which is the function shown here: alpha, delta and epsilon. The replication of non-damaged cell in the nucleus during the S phase, it is involved in the process of replication, and gamma is responsible for mitochondrial replication, and additional enzymes are available that includes eta, kappa, epsilon and theta. They are responsible for replication of damaged DNA, and totally we have alpha, beta, gamma, epsilon, sigma, lambda, mu, pi, psi, pi as well as theta. They are referred in the DNA repair mechanism, that we will see the role of the DNA repair in following our video. These are characteristics of eukaryotic DNA polymerase. Especially, as I told you, 3 polymerases are associated with our nuclear DNA replication, that is alpha, delta as well as beta, and one is involved in the mitochondrial replication, that is nothing but our gamma. And here the table shows the comparative detail of our various DNA polymerases. As I told earlier, the DNA, since the genome size of the DNA is very larger and number of chromosomes also more in the case of the eukaryotic system, especially in the case of the human, it is very difficult to start replication from one end to the other end, and it will take longer time to complete. So nearly 6 to 8 hours it takes to complete copying of one set of chromosome. So eukaryotes have longer and linear chromosome. It requires multiple origin of replication. Already I gave you example. The replication begins at various points like an NH containing bridge construction at various locations, and they are connected together to ease the transport. No, same manner it is done at several places, and they are connected with each other by the DNA ligase enzyme. This was identified by Huberman and Riga during 1968, and moreover the DNA replication is bidirectional replication, and here is the photographic evidence which shows the chromosome, what is the centromere, where is the origin, and multiple origins are available, and the DNA is synthesized during the S phase of the cell cycle, and it comes to an end in the synthesis of our chromosome itself. So both the sister chromatids are copied or sister chromatids are copied by the process of our replication. This is a micrograph of a replication which is showing multiple origin of eukaryotic chromosome. The origin site of eukaryote has some similarity that of our prokaryotic system. Here is an example of Saccharomyces cerevisiae, that is a yeast that is shown as an example, which is looking similar to that of bacteria. We call that origin site containing elements are called ARS elements, that is autonomously replicating sequences, which is similar to 13 base pair repeats and 9 base pair repeats of our prokaryotic system. The ARS elements are about 100 to 150 base pair in width, and moreover it is highly rich in AT sequence like our 13 base pair and 9 base pair repeats, and these are there are 3 to 4 copies of the specific sequence is available, which is similar to the DNA box, that is 13 base pair and 9 base repeats of our prokaryotes, that is bacteria. In the origin site or autonomously replicating sequence, that is ORI site of eukaryotic system, the replication begins with the assembly of various set of factors. That is, it initially forms a pre-replication complex, we call it as pre-RC. Totally 14 different proteins and are involved in the process of a pre-replication complex formation, and 6 of them are called origin replication complex, that is ORC, we call it as. ORC means origin replication complex. When it commences means the replication, the assembly of origin recognition complex takes place during the G1 phase of our cell cycle. Other pre-replication complex binds to the MCM helicase later. Here is the stepwise mechanism how the recognition of origin is taking place, that is ORI site is taking place, and what are all the steps or what are all the proteins that are associated, that we will see one by one. Origin recognition complex binds to the origin of replication, and a mini chromosome maintains this maintenance protein, that is MCM is binding with ORC. CDC6P is required for binding of MCM as well as ORC. The initial complex is activated and the helicase activity opens the parental strand to form the small bubble, because the double helical DNA will be twisted around each other just like a plate what we have. So it has to be separated to open it. Same manner here, the binding of this MCM with ORC with the help of a CDC6P responsible for the activation of helicase and unwinding of the DNA, that is separation of the DNA. It breaks the hydrogen bond between the parental DNA, double helical DNA, and it gets separated into small bubbles. So the replication, once it is opened, the single stranded DNA is exposed to damage. It may be attacked by any nucleases, so that the DNA will be damaged. To avoid that, we have single stranded binding protein, that is replication protein A is come and binds to the single stranded region. So in the prokaryotic system, we have single stranded binding proteins are there. Similar to that, we have this replication protein A comes and joins. Then now the DNA polymerase A, that is alpha, that is associated with primase, that comes and joins to this position. Once the DNA polymerase alpha and primase complex joins, it synthesizes a short RNA, that is RNA primer, which is and forms a DNA RNA hybrid. Normally the length of the RNA nucleotide will be followed by initially it will be 10 and 20 to 30 nucleotides are synthesized. Once the DNA alpha primase complex leaves, then the DNA polymerase containing our delta and epsilon proceeds the elongation of the leading and lagging strand. After the primer is removed by RNase H, and in the case of the leading strand as well as lagging strand, and the ribonucleotide attached to the DNA is further removed by flap endonuclease 1, so that the process of replication gets over. So we will see how the in detail steps are associated. So as I told you, the eukaryotic replication occurs in the S phase of the cell cycle. The replication occurs only one in a time a cell. The replication occurs in 5 stages, whereas in the prokaryotic system it is only 3 stages. Here it is 5 stages: pre-initiation, initiation, elongation, termination, and telomerase function. Here we can see that the DNA pre-replication complex assembly, and once it is assembled, then G1 cyclin CDK inactivates CDH1, then G1 cyclin CDK activates the expression of S phase cyclin CDK components. Then further, the fourth step, G1 cyclin CDK phosphorylase S phase inhibitor. Then the fifth step, the SFC proteasome degrades the phosphorylated S phase cyclin CDK inhibitor, and the S phase CDK cyclin activates, and the pre-replication complexes are formed for the process of our DNA replication in the synthesis. Once it is over, then comes the mitotic cyclin CDK that activates the early mitotic events, and further it promotes the process of our movement of a cell cycle from, or cell division from, S phase to metaphase, then followed by anaphase, and finally the telophase and cytokinesis happens in the case of the cell. So just it is moving from the S phase to G2 phase, then in the G2 phase, from the G2 phase only it moves to the cell division process. It goes to metaphase, anaphase, and telophase. Again the cell, after the cytokinesis, telophase and cytokinesis is over, so that is cell division is over, then it moves to the early G1 phase. Then it is taking a rest of G0 phase. Then when there is a need only, again the G1 phase gets activated to G1, early G1 and mid-late G1. Then it moves to S phase G2 mitosis. The process keeps on going in the case of our cell division. The replication selection is normally mediated by, already told you, pre-replicative complexes. The first step is the formation of the pre-replicative complex at the site of replication, that is origin complex. This is associated with two helical loading proteins, CDC6 and CDT1, comes and joins, and further in the replication region, fork region, another enzyme comes and joins, that is nothing but your helicase M227 complex. This comes and joins at the replication origin site, that is at the bubble region, we call it as. The pre-replication complexes are activated at the second stage of our replication, that is initiation. It initiates the replication by two protein kinases, namely cyclic dependent cyclin dependent kinases and CDK, that is CDT4 dependent kinase. What is kinase? Kinase is responsible for the addition of phosphate group to the target protein. Here what is a target means? Here it is phosphorylating two proteins, that is your G1 protein, which is entering into our cell into S phase. So the phosphorylation of the protein further results in the assembly of additional replication proteins at the site of origin and to start the initiation of our replication. Once the initiation process containing G1 phosphorylation is over, then comes the three eukaryotic polymerases are recruited at the origin of replication at multiple points. As I told, multiple origin of replication, bubbles are there where these processes are carried out. The DNA polymerase delta and epsilon associates first and followed by DNA polymerase alpha and primase. The order of these all these polymerases are present at the origin prior to the synthesis of first RNA primer. Normally the primer is synthesized by the DNA polymerase alpha, that is primase. Once the primer is synthesized, then comes the role of our polymerase delta and epsilon. Thus the initiation of replication gets started. So here we can see that the diagram, the primer synthesis by our DNA polymerase alpha, it provides a short segment of primer about 20 to 30 nucleotide length in the both leading as well as lagging strand. Once the primer is synthesized, the DNA polymerase epsilon is recruited and delta is also recruited for the process. Epsilon is responsible for our leading strand synthesis, whereas delta is responsible for our lagging strand biosynthesis. The elongation process is the third step of eukaryotic replication. The resulting primer template junction is recognized by the eukaryotic sliding clamp loader, which resembles that of our sliding beta clamp of our prokaryotic DNA polymerase 3. So this is called as PCNA. PCNA means primer associated nucleotide clamp, sliding clamp at the sites. Then now the delta and epsilon recognizes the primer and begins the synthesis. As I told, the epsilon is responsible for our leading strand production, whereas delta is responsible for our lagging strand production, which is shown in the diagram. Once the leading strand and lagging strand synthesis is over, the movement of the enzyme takes place and looping up of DNA occurs, and further the extension, that is helicase, is responsible for unwinding of DNA in the forward direction. Then the enzyme is relocated in the case of the lagging strand to a new position for the process of our new primer synthesis in the lagging strand. So further the role of delta takes place for the elongation purpose in the primer region. Once this is over, after a few lagging strand template containing fragments of DNA is synthesized, then comes the role of removal of primer and filling up of the gap in the case of the eukaryotic system. The RNase H is responsible for the removal of RNA primer in the case of the eukaryotic system, and once it is over, it is like our DNA polymerase 1 nick translation process. So once the RNA is removed, the gap is filled, and again the sealing is taking place with the help of our DNA ligase enzyme. Once the replication fork meet with each other, the termination process occurs. The formation of duplex DNA happens in the case of the system, and a short amount of nucleotide will fall off without completion in the case of the eukaryotic replication, because it's a linear DNA. This shortage of DNA is in turn corrected by telomerase enzyme. That is, in the leading strand it keeps on continuing, whereas in the lagging strand what happens, since it reaches the end, when it reaches the end, the last region or the end of the DNA, that is tail of the DNA, the chromosome, where the Okazaki fragments will be initiated in the lagging strand few nucleotides prior, but the gap will be there. That gap has to be filled in the case of our system, or else in every round of replication what will happen, the nucleotide will be lost and the size of the chromosome will keep on reducing, so that the problem will be there in passing the information. And one fine round of replication, what will happen, the number of nucleotide will be lost in the chromosome, the shortening of the chromosomes will take place. To avoid that process and to guard our end of the DNA, that is telomeric region, telomerase enzyme comes for the rescue. That is, linear chromosomes have telomeres at both the ends. The term telomere refers to the complex of telomeric DNA sequences. It's a repeated, it's a moderately repetitive random array of TTAGG. This is the telomeric sequence that is added by the telomerase enzyme. It adds to the overhang region of our 3' end of the eukaryotic chromosomes. Normally the overhang, how many nucleotides will be overhang, means nearly 12 to 16 nucleotides will overhang. When we leave it as such without the addition of our any telomeres or replication process, what will happen, this every time the 12 to 16 nucleotides will be lost, the length of our chromosome will be shortened. One fine point, no nucleotides will be there in our chromosome. To avoid that, to safeguard the ends of the chromosome and to copy the entire nucleotide without missing even a single nucleotide, what it is doing is, it is making use of our telomerase enzyme. So in the case of the telomeric repeats, when it is added to the ends of the chromosome, it varies, and the composition of the telomeric sequence also differ from one eukaryotic system to the other eukaryotic system. For example, in mammal or human, we have TTAGGG, that is 6 nucleotides are found, whereas in the slime molds it is TTAGG, and Dictyostelium it is AG 1 to 8 repeats, and filamentous fungi, that is Neurospora, it is the same like a human being, whereas budding yeast Saccharomyces cerevisiae, it is TG 1 to 3 repeats will be there, and ciliates Tetrahymena, it is similar to human, and in Paramecium, in addition to in Paramecium, TT, G, triple G and T or G will be there, and Euclodes, it is 4T and 4G is present, whereas in higher plants like Arabidopsis, we have TTTA, that is triplet of TA, triplet of G. So this is the telomeric repeats which are found at the end of the chromosomes in the eukaryotic system. So, and since the polymerase persons two unusual features. They synthesize DNA only in the 5' to 3' direction. There is no place for the 5' primer to add the nucleotides. This is a problem. That issue is created why it is not synthesized. This is a diagram which shows what is a gap, how it arises during the replication process. How it is to be solved. The linear chromosomes become progressively shorter with each round of replication. To solve this issue only, the DNA sequences are added to the ends of the chromosomal DNA, that is telomeres are added. It's a special mechanism catalyzed by the telomerase. Already I told you, telomerase is an enzyme that contains protein as well as RNA. This RNA of the enzyme acts as primer. It is this enzyme is also called as inchworm polymerase. The RNA is complementary to the top DNA sequence found in the telomeric repeats. This allows the telomere to bind with the overhang region of 3' end. Here we can see that the eukaryotic chromosome and a gap is there. This RNA is complementary to that region. It comes and joins, and the telomere synthesizes the 6 nucleotide repeats, and further it lengthens. Further it moves 6 nucleotide to the right and begins and makes another repeat. Once the complementary strand is added, the primase comes and joins, and the primer is and the DNA polymerase comes, and it extends the RNA primer and fills the gap, and the ligase comes and joins. Here you can see that how it is extended further. The process continues until the number of repeats which is found in the case of the system. In the eukaryotic system, telomeres are playing an important role in protecting the ends of the DNA. In every round of replication, to avoid the loss of nucleotide like this overhang, what it is doing, it is adding number of copies of our telomeres to the end of our DNA. That is the significance of DNA. It is protecting the DNA in both the ends and prevents the loss of genetic information from parental DNA to the daughter DNA. There is another significance associated with telomere is that as we age, the number of repeats of telomere declines. So as we age, the age of the cell can be identified with the number of telomeres present at the end. That is first significance. Next significance, in the case of the normal cell, it will be retained as such. Once there is a need only, the cell will divide and the replication starts, and the telomeres are added to the DNA. But in the case of the cancerous cell, what happens? It is an uncontrolled cell division takes place. In order to cell has to divide, the DNA has to be copied. We are seen in the cell cycle. So the DNA has to be copied means the number of repeats that is added will be lessened during every round of replication in a cancerous cell. When there is a drastic change in the level of DNA repeats, it is an indication for cancer development. So it is identified and confirmed that the cancerous cells containing number of telomeres repeats are lessened when compared to the normal cell. And that's all about our overall mechanism of eukaryotic DNA replication. Thank you.